Can do the second half. So my ambition now, after this very basic biological course, is to show in a few still superficial approach how our crowd, what the structure which made us human, and all this great intelligence. We are so damn intelligent. This is the cerebral cortex, which made us to be a human being. So how this extra complex, beautiful sheets of the cortex is developing, the sheets of cells developing from scratch. Because there is an age, I will show it to you, there is no cortex whatsoever. So the development is not like you have a small cortex which will grow, there will be a big cortex, but there is no cortex and there will be a cortex. Okay? So what I thought that first, before discussing all these steps, why not just have a quick portrait of the cortex? Why is it so interesting? So first of all, look at this sort of artistic representation of the cortex. So this is the way, in my opinion, this is the way you should imagine the cerebral cortex. That is, this is a huge sheet. Huge. Look at this. 2,200 square centimeters. That's really a fantastic surface. Okay? Which just housing 10 to the 10 neurons. That's an incredible number, okay? And if you look at the evolution, the major event during evolution is not that your nucleus caudatus or the thalamus, which really growed compared to the other animals, but the cortex made an extraordinary development. If we compare, for example, the ratio of the cortex towards the rest of the brain, this ratio we can take in the mouse 1, and look at this ratio is increased 10 times with the monkeys, and there is 100-fold increase between the human and the monkeys. That's extraordinary. And just why do I show this picture? Because in fact, not only the volume of the cortex increased during evolution, but the surface. You know, if we consider the thickness of the cortex, that's not a big deal compared to the mouse. Really, this major event of evolution of the increasing size of the cortex corresponds to the increasing of the area, the surface. This is the parameter which is just drastically changed in the human being. So now you ask the question, so we have 1,000 times increase compared to the mouse. So we are just a big mouse or something that's changed during evolution. So let's have a small look at. I will not give you definitive answer, but it's so interesting to scratch, to see what is behind of this increasing surface. So what? The first thing which is important is if we compare the mouse and the human brain, everything which is colored we call the primary cortices. Everything which remain white are the associative cortices. Okay? Two different things. Primary cortex, we have a primary motor cortex with which we are moving the muscle. We have primary sensory cortices, some of the sensory, which is the station in the cortex which receives all sensory information. Primary cortices are the direct station of the cortex and the subcortical crunch. The associative cortices are everything which is important in our life for all the cognitive functions. Now look at the ratio. Look at the ratio. 2/3 in the mouse are primary cortices. 2/3. Well, look at the human brain. There is a huge expansion. I would say if we accept this increase in surface area that corresponds to the associative cortex. Okay, a huge increase in association areas in mouse, in human compared to other animals. Okay, now we accept it, that's fine. Now we understood something more. Just, I tell you, 30 years ago, most of this associative cortex had no function. So even people wrote in books, they're saying that most likely this is just an evolutionary access. And we can live very well without these associative cortex. 30 years ago. Okay. Now, so what can we gain with this increased surface area, right? Well, put it in a very simple way, there is an explosion of the number of specialized areas. Okay, shown here, which is of course it's a sort of humanistic representation that how many individual specialized area we just get with this increase in the area increase of associative cortices. It's incredible. Okay, just look at the visual cortex. All these sub-regions, all these specialized region which has a specific function for color vision or whatever integration. This is what we gain with the increased surface area during evolution. With these pictures, I want to go some boring parts. Okay, what can we gain at the level of the structure? What with this surface increase? So, is it possible that humans and mouse has a fundamentally different cortex? Well, let's see. So this is a histological picture. Remember, the definition of the cortex is the layered organization. So in the neocortex, we have a 6-layer superposed organization. Layer 1 is the most exterior towards the surface, and with layer 2, 3, 4, 5, and 6. If you don't see clearly the 6-layer, it's normal, of course. You have to imagine everything in this histological picture. You have to imagine things. So more or less, we have the 6-layer. Is it different in human and mouse? Not at all. Not at all. Really not. So we have the same. We call this horizontal organization of the cortex in the mouse and the human brain. Okay. So that's not it. Let's see. We have also a second type of organization, okay, in addition to this horizontal layer, like organization in this millefeuille, the cortex, okay. We have a sort of vertical organization, so perpendicular to the surface. So we have, this is what we call the columnar organization, okay. So these are sort of repeated units in the cortex. This is the surface, you see. Like this. So look at the mouse. They have about 1,000 of these cortical columns, or the poor mouse. And we have in the human brain, we have about 2 million of these cortex. So increasing the surface area produces a huge expansion of the number of vertical units, columnar units. That's a quantitative difference compared to the mouse. Do we have a difference in cellular composition? You remember the cytology? The basic composition in mouse and human is exactly the same. We have glutamatergic excitatory neurons, pyramidal cells, and we have GABAergic interneurons with a huge diversity. Do I want to say that we don't have new cell types in the human brain? No, we have. We have a few different cell types. It's not today that I will talk about it. Yes, neuronal diversity in the human brain is increased, but that's not a big deal. For me, the big deal is this. If you increase the surface, then you increase drastically the number of this vertical unit, and then look at this. Some people 30, 40 years ago, they took the time, they counted the synapses in the human cortex and in animals too. what they discovered is this. 90% of all connections in the cortex is in fact coming from inside the cortex. 90% or more than 90% of the connectivity is cortico-cortical and not with the other subcortical structures. This is amazing. So the intracortical connectivity is the dominant sort of network configuration. Now you can see something very clearly. If you increase the surface of the cortex, you increase its vertical unit, and then most of the connectivity is cortico-cortical. Then what you do is you expose the synaptic network, the possibilities, the complexity of cortico-cortical or intracortical connectivity. It's just an explosion due to this expansion of the surface. Okay, so this was my personal portrait, just to show what is really specific or interesting in cortex. Now the question, how we can create during embryonic development such a crazy complex, beautiful structure. So here is the timeline, more or less, which is true for the brain and for the cortex. And I represented here three curves, three major events in the creation of the cortex. And this is valid for your other courses, which won't be given by me. So first, you remember the layers, okay, horizontal organization. After conception, the first wave of events is the layer formation. And this is what we will discuss today, layer formation, because this is linked to two major events, neurogenesis and neuronal migration. In the second slides, I show you the video time-lapse of the second type of migration, that is axonal and dendritic growth, you know, the firework. Okay? Well, all these fibers will find tentative or potential targets, partners, okay? We call it pre-wiring. This is all embryonic events. And then I will show you more details, but then look at this. Just around birth starts a new period, which lasts even in postnatal years. That is what we call activity-dependent plasticity, refining the pre-wiring network, okay? This is the three major events. And now we're going to concentrate on the very first studies of events, neurogenesis and neuronal migration. 38 weeks of gestation, right? Intrauterine life. And I want to guide you somewhere here. Week 6. The human embryo has 13 millimeters, 13 at this stage. This is the shape, okay? And he has a brain. He has a brain, okay? This is the neural tube, you know, this is the telencephalon and all the other part. But this embryo has no cerebral cortex whatsoever. 0. 6 weeks. Okay? This is really interesting. So, at this time point, the 6 weeks, somewhere here, there is an extraordinary speed of proliferation of neurons. That is to say, every single minute, 250,000 new neurons will be created in the germinal zone of the ventricular wall. Okay? Everything starts in the ventricular wall. I will show it to you. This speed is absolutely extraordinary. Proliferation, generation of corticoneurons, they calculated 250,000 new neurons every minute. Imagine what's going on. It's extraordinary. So then this goes on, and somewhere here, okay, somewhere here the fourth and the fifth month, this proliferation stops. So the maximum number, what it means, the maximum number of cortical neurons is reached during the first 5 months of our intrauterine life. Okay, and then this maximum number will just decrease further on. Okay, 5 months and we have all the cortical neurons. Now let's see what happens with these millions of neurons generated in the germinal zone of the ventricular bone. So here is an image, a sort of coronal image showing the telencephalon, okay, the forebrain. We have the 3 ventricles, ventricle 1, ventricle 2. Within we have the plexus choric, and this is the 3rd ventricle. As I told you, the proliferation will start in the bone. Generation of new neurons, the neurogenesis is in the bone. You know, if you imagine that it's like the Mediterranean. Okay, all the culture is coming from the Mediterranean. All the neurons coming from the wall of the ventricle, from here. Now this is a scanning electron microscopic picture of this extraordinary proliferation phase. The ventricular zone, you see these progenitors and stem cells producing new neurons, as I told you, 250,000 every minute. What is important to realize that we are exterior to the future cortex. This is another place. This is the wall of the ventricle. This is not the cortex. So what happens is, if we imagine that this is the ventricle is somewhere here, this is the surface. So neurons must migrate from this ventricular germinative zone towards the future cortex. So 2 events will precede this extraordinary migration. And now it's important also to emphasize that as I showed you the video time-lapse, I will use radial migration as a principal subject today because it's so interesting and we are not talking about other. So I am talking about radial migration, and I'm talking about pyramidal cells, only pyramidal cells. So layer formation is the production, migration, and positioning of pyramidal cells. Okay, extracellular pyramidal cells. So how pyramidal cells know where to go? How this migration is guided? What is the mechanism? 2 events will precede the migration. One is the establishment of these crazy simple cells. As I told you, the grandmother cells, radial glia, it's a scaffold. At the same time, it's a stem cell, as I told you also. So this scaffolding cell will establish a stretch between the ventricular zone down here and the surface of the cortex, where the future cortex will be here, somewhere here. Okay. The second event, which precedes the migration, is the positioning of this cell, which is called the Cajal radial cell. Sorry, it's in French. So Cajal radial cells. Strange name, if you wish. No, you have to learn. Good. Okay, so this is the 2 events: radial glia, scaffold, and Cajal radial cells. And then, as you see, we call it PP, pre-plate. Pre-plate will be splitted during development. Cajal radial cells will establish there. And between the marginal zone, which is the surface, and the subplate, okay, it doesn't matter, these small details I will not ask, the future cortex will be generated step by step, layer by layer. How? So here this fluorescence image shows, for example, the migration, what I showed on the video. Okay, this is the ventricular zone, the guy was multipolar and then get very elongated and migrated along radial glia towards the surface. Good, this is what we have seen already. Now something crazy in the cortex. You remember 6 layers in the neocortex, number 1, 2, 3, 4, number 1 is the most exterior. So what would be logic for you is that, okay, we have a production of neurons in the wall, then all these neurons migrate towards the surface. So what would be logical for you is that first we have layer 1, right? Established, that's fine. Next, layer 2, next, layer 3, 4, 5, and 6. That would be an easy job, no? Just establishing sequentially, but it's absolutely not the case, and we just don't know why. So what happens is that the first wave of cells will be proliferating, generating the first wave of neurons. You know what type of neurons? These are layer 6 neurons, the most inner layer, layer 6 in red. Okay, next wave, well, that will be layer 5. What is the consequences? That layer 5 neurons have to go through this already established layer 6. So that's a complete fight to go further and just bypass the already established layer 6. And then layer 4 cells should do the same thing. So first they have to go through layer 6, layer 5, and then establish layer 4 cells, and so forth. We call this sort of generation of our beautiful neocortex the inside-out constriction. Don't ask me why, we just don't know why. It's going like this in a very complicated way. Inside-out construction. So first layer 6, the most inside layer, and then progressively the others. Yeah, sure.
In the previous slide, you told us that.
Do you hear her?
In the previous slide, you told us that the progression starts at the wall, but now we're talking about an inside-out growth, and I'm kind of confused. What do you mean by the wall then? How come it's not outside?
Okay. The wall is the ventricular wall, it's right here, okay, which is represented here. It's always, this is the wall of the ventricle, you know, the cavity. Every neuron is generated here, and they should migrate. And why we say inside? Because layer 6 is the most inside layer. Layer 6 first, layer 5 after, so it's always coming from the wall. But the most inside layer is generated first. So this is why we say inside out. That is to say first inside, then the outer layers. Good. Okay. It's a complicated issue. What is really important is that every layer has a different genetic makeup, a different transcription factor makeup, and so timing will determine all the phenotype of these different layer neurons. So the timing of the birth and the subsequent migration is the two major events which generate layer. So when layer 6 are migrated, then layer 5 neurons started to be generated by the stem cell, the cell souche, okay, and so forth. So we have a timing, a wave of generation and migration, and this is basically what creates the 6th layer in a sequential way during 3 or 4 months. The whole cortex is generated from scratch. Okay, so it just want to show that specification is time dependent. For example, once you made layer 6, you cannot make again layer 6. Okay, the next is layer 5, etc. So the timing is absolutely important in everything during development. Okay, so the birth rate of neurons determine the laminar specification of the cortex. You cannot make layer 6 cells 1 month later. It's not possible. The stem cell is changed and we generate only the superficial layers. Okay, same thing. So with each different type of cells with different layers, this is phenotype. This was the first code, remember? So with this sequential production, we have a sort of fingerprint of transcription factor, genetic makeup of every different type of neuron. Okay, I pass through. Now a few words about radiogram. As I told you, these are very simple cells. Boring. These are boring cells. Just scaffold cells. This is the ventricle, this is the surface. We are stretching from the ventricle to the surface. It's as you understood, the main role of radial glia is guiding the migration. Okay, guiding the migration, the radial migration of neurons. Okay, so here is how you have to imagine this event. So we have a sort of, in the ventricular wall, we have a blueprint, sort of, we have a blueprint of the future cortex. So it's like a mosaic. And each small area of this mosaic is linked to the future cortex by a radial line. See? It's an interesting concept, right? We have a design, a blueprint in the ventricular wall, and then neurons are produced in these mosaics, and they will be guided to the cortex on that mosaic too by the radial line. Okay, you understand? This is what we call the proto-map hypothesis proposed by Rakic. Proto-map, that is to say, there is a proto-map in the ventricular wall. And there is a one-to-one correspondence between these mosaics in the ventricular wall and the future cortex. Remember, the columnar organization, that's the way you should imagine these mosaics in the cortex, okay? So that, here you can see again, column in the cortex, the unit, the mosaic, the protomap in the ventricle, and here you generate the cortex, right? Now people were really crazy about to understand why we do have this expansion of the surface. Okay, so they made mutant mice in which they overexpressed the beta-catenin. It's a signaling, it doesn't matter for you, really. Beta-catenin is the main player of Wnt signaling. Wnt is so important, very powerful, very complex signaling, which is involved in organogenesis, cancer, regeneration, etc. WNT, Wnt signaling. So here they overexpress because they said, okay, maybe this Wnt signaling proliferate more, so from mouse we can make a huge brain, human brain. Okay, look at it, what happened. So they created a sort of G-fied mouse brain, which is quite, this is the control, this is the transgenic mice. Well, the only problem that the mice cannot survive. After a few days after birth, the whole brain was just disintegrating, so it didn't work. But it's a really interesting question. What happened during evolution? Why do we have this expansion of the surface in the human brain? Okay, now recently they found something which is really, really interesting. I don't want to present this as the only explanation why we do have a huge surface, but normally in the mouse brain we have radial glia, okay? Stem cells, progenitors, linking the ventricular wall to the surface. You understood, scaffold. Now what discovered in the human brain, which is almost absent, there are very few cells, but almost absent in the mouse brain, is what we call the ORG, outer zone radial glia, which is really interesting. Why? Because there is an increased number of these cells in the human brain. We call it the outer subventricular zone, that's just over here. Okay, and what is characteristic for these cells is they have a link with the surface, but they do not have a link anymore with the ventricular surface. And these guys are proliferating. So one idea is that in the human brain, okay, the extreme proliferation of these outer zone radial glia cells will give an expansion of superficial layers, increasing this, thereby the surface of the human brain, which is absolutely not the case in the mouse. We don't have, we have very few of these cells in this outer subventricular zone. So this is just one explanation. The message for you is that we just don't have the slightest idea why this beautiful cerebral cortex in the human develop. But there are ideas and there are some explanations. Very good. So as I told you, and I showed it to you, radioglia, everybody was surprised that this crazy boring cells is the stem cell in the human brain and in the mammalian brain also. So radioglia can give rise to neurons. This is what happens during these 5 months in the human intrauterine development, okay? And what is interesting that this is a stem cells, but during this proliferation, this stem cell is continuously changing. It means that at the beginning of the neurogenesis, they are able to produce layer 6 cells, but 1 month later, the same cell cannot anymore produce layer 6, but layer 4, or layer 3, or 2. So there is a progression. Radioglia is changing all the time. And the bad news for us, at least for the moment, is that around birth, these cells are unable to produce any more neurons, just glia cells. Okay? So there is a switch in neurogenesis to gliogenesis. Our beautiful multipotent stem cell is changing the potential, reducing the potential, unable to produce neurons, as I showed you, except 2 regions, the dentate gyrus in the hippocampus and the lateral wall of the ventricle, sending cells to the olfactory bulb. So switching from neurogenesis to gliogenesis and no return. Okay, and this is, if we are talking about this event, migration and neurogenesis, gliogenesis is a late event. Practically we don't have, except the radioglia, we don't have in the brain a glia during this first period. And gliogenesis starts to grow up here, and here you can see this curve representing gliogenesis during the coarse wiring state. Okay, good. Now, a few words. So, you learned at the first part of my talk that migration is an important process. There is also machinery. The most important is this cell adhesion, and indeed, these migrating cells, they are sticking to the process of radioglia. Okay, so they're climbing up. Adhesion is a very important event. I showed you also how cells can move, cytoskeleton, actin, neurotubulin, and then I also showed you that soma translocation, nuclear translocation linked to the cytoskeleton, pulling force of microtubules, the centrosome, etc. is very important. Now you understand that these events are very precisely coded during development. Any kind of error, that's fatal. Okay, let's see what happens if there is an error. So for example, we can have mutations, spontaneous mutation in babies of cytoskeletal molecules in the centrosome, which just completely handicap cells during migration. This is what I showed you here with the first slide. And then the consequence is tragic. If we have a major mutation, for example, in a gene which is called LIS, okay, or other cytoskeleton or centrosome related molecules, then we have a lysencephaly. That is to say, no gyrification, no surface decrease. That's a tragic situation. Babies has a smooth brain, lysencephaly, okay? When this is not a widespread phenomenon, just few cohorts of cells are late or lost during migration. The consequence is that we have heterotopias. Okay? Heterotopia meaning mispositioned neurons. They are lost during this long trajectory of migration. They are either badly guided or somehow they are slowed down. They are unable to move forward. So then we have misplaced cohorts, groups of neurons within the brain. That's also very dangerous because in many cases of these heterotopias, in fact, 20% of child epilepsy is linked to this heterotopia. That is to say, these neurons will make crazy contacts excitability of these heterotopic groups could be very high, and very often is linked to epilepsy in childhood. So heterotopias are also very important pathologies. Now, let me just show you one example. About 30 years ago, they discovered that there is a mutation. We call it the mouse, the real mouse, in which the gene, the reelin, has been mutated. It doesn't matter the details. So they noticed that these mice become epileptic, they have motor problems, and their cortex was inversed. What does it mean? You remember I showed you the inside-out construction of the cortex? Now this inside-out construction was blocked because of this mutation. This is it's a button-like protein. And the cause, we call it the Norman-Roberts dysencephaly, and the mutation was the chromosome 7q22. Okay, let's see what happens, because this helps us to understand something in this migration business. So what happened is, here you see the normal development, as I told you, that there is this splitting of the preplate and the future cortex is placed here, and the successive waves of migrating neurons, they are generating a sort of inside-out cortex. Now, what you can immediately see, when this in realer mice, when the protein is lacking, then, very simple, the preplate cannot be splitted, so the cortex started to be accumulated, cortical cells behind, and in fact they observed directly in the video time-lapse microscopy that layer 5 cells cannot move further than layer 6. They are unable to bisculate, to go further, and so the cortex will be inverse. Layer 6 will be the outer layer, and then layer 5, 4, etc. Okay, this is what happens with it. So what they realize, yes?
Is the cortical thickness different with the real mice or not?
No, not really.
Okay.
Just completely disordered. Okay, that's a mess. Okay, so here is what they discovered. Really, the protein, remember I told you, but I didn't tell you why is it important. I told you that two events precede the migration. One is the radioglia, and second is the Caha-Retius cells position. Caha-Retius cells is positioned towards the PL surface, the marginal zone. And what they do, these cells, they produce really the protein. So what is the role? What is the role of reeling during migration? Why this chaotic situation established where reeling is not there? Well, we learned the following. Caha-Retius cells produce and establish a concentration gradient, meaning that it's very high here, close to the surface. This black guy is the Caha-Retius cells. The red crosses is reeling. So a very high concentration close to the surface, and then there is a decrease in concentration when we go inside. Now what happens is that cells generated in the wall, they kept even the lowest signal of reeling deposited by the Caha-Retius cells. So migrating cells will cap this reeling signal. And so at this point, the multipolar cells, the freshly generated new neurons, will be polarized. And then these polarized cells will attach to the radioglia. And then the cell will migrate towards the surface, following the gradient, the concentration gradient of reeling. So two major effects of reeling, very interesting. One is the polarization, and second, you remember I told you, attraction. So maybe you ask the question, how come that our cells, our pyramidal cells are migrating in radioglia? That's fine, they are guided, but why don't they turn back? Why? Because reeling is there. Not only they polarize these cells, but also reeling, the concentration is attracting these cells towards the surface. So they absolutely don't want to turn back. They are attracted. The growth curve is capturing these messages that, okay, move to that direction. And this is what completely disturbed in the real. Okay? So now you understand why we do need the Caharetio cells, why we do need the radial glia producing new neurons, guiding the migration. Very good. Let me, so more or less, this was the story of pyramidal cells. You understood really interesting things, that there are waves of generation and migration of neurons. Layers are established. Okay, there will be pyramidal layer 1, 2, 3, etc. We understood that there is a guidance, direct guidance by radial layer, also attraction by the real. But if we want a good cortex, not epileptic, because all these pyramidal neurons are excitatory neurons, we need GABAergic inhibitory cells. Curious things, GABAergic cells also generated more or less the same time, but they don't give a damn about the radioglia. And also, while the pyramidal cells are produced up here, on the wall of the ventricle, GABAergic interneurons are produced down here, in the ventral pallidum. Okay, a completely different way. Isn't that interesting? The cortex is composed, very important cell type, GABAergic pyramidal cells. These two types of cells are generated in two different places. And I will show you now that these cells are also migrating, but in a completely different way. Okay, and they will meet, and finally, they will generate a beautiful network in our cortex. So let's see what is the difference between the radial migration, what we learned, and the GABAergic migration. This is wholly complex. I don't have to I would like to show you another principle of guidance for the GABAergic cells. So these guys, GABAergic neurons, must migrate in this way, parallel with the surface, not perpendicular as the radial glia guiding migration. Parallel, okay, from here in the ventral forebrain up into the cortex. We call this migration tangential migration of GABAergic neurons. The region which produces these GABAergic neurons called ganglionic eminence or subpallidum. Okay, I don't want to talk about this now. Here is the interesting part. So these neurons are coming from this ganglionic eminence, and here is the future striatum in the same region. People get crazy to observe, migrating these cells, what they observe, that all these future cortical GABAergic neurons pass in this direction, and they don't give a damn about the future striatum. They never migrate into, never. They just continue the long distance migration up here into the cortex. So they ask the question, how come that these neurons strictly avoid the future striatum, although there are GABAergic neurons in that circuits too. There is no circuits in the brain without GABAergic neurons. So they ask the question, what is that which makes these cells so disciplined, migrating tangentially up here in the cortex, never entering this region? Just to cut the long story short, they discovered that striatal cells produce negative, repulsive factors, and namely the semaphore. Okay? Remember I told you, interesting the name, the stop signal, the semaphore. So striatal cells produce this, and the migrating cortical neurons coming from here will detect these negative signals, and this is why they will avoid entering into the future striatum. So what I showed you, this principle, as opposed to the radial glia guided migration, is what we call the guidance by permissive corridors. Okay, guidance by permissive corridors. This corridor is open and is positive. There are positive molecules which attract GABAergic neurons. Doesn't matter the name, neuroregulin or another chemokine. But the concept is very interesting that this corridor, left and right, is paved by the semaphorin, the negative factor. So this guy will continue going up to the neocortex. So this was the principle of tangential migration. Semaphorin was one place. And now I would like to show you if mentally you can stay ready for a few minutes to follow me. This guy, Efri, this is also really interesting. Otherwise I would not tire you with this last few minutes. So Efrin is again, one more time, a sort of repulsive one, a stop signal. And its receptor is called EPH, okay? It's not the name which is important, I just want to show you some interesting, what the heck I want to show it to you, okay? I don't care, okay, yeah, look at this red line. This is the generation and migration of pyramidal cells. And look at this black line here, just to illustrate that GABAergic migration, tangential migration, is an ongoing process, even around birth. This is really important. That is to say, establish the excitatory neuron architecture of the layers, and then we will insert new and new GABAergic neurons according to the needs to get an equilibrium, inhibition, excitation. So GABAergic migration goes on even around birth, while pyramidal migration and generation is a very early event. Okay? This is just a paper in which they show this crazy migration even in babies, newborns, of GABAergic cells. And now, okay, as I told you, guidance, you understood the mechanism of migration. Now I'm illustrating the guidance. The first example was radioglia guided migration. That's a direct guidance. Then I showed you directional guidance by permissive corridors, GABAergic cells. And now this is the last one, guidance by gradient. If you ask me what is really outstanding about the cortex or what is the main function, in fact, this crazy complex sheet, the cerebral cortex, is making maps of the outside world, the visual world, the auditive world, your body, everything is mapped on this screen, the cerebral cortex. This is one of the most important function of the cerebral cortex, generating organized, topographically organized maps of the environment, the outside world, and our body. Now, of course, biologists, when they see how the heck this complicated, topographically organized map is generated during development. Okay? So Roger Sperry, in 1943, they put forward the chemo-affinity hypothesis. He said, it's not possible that if you imagine a map that's a 2D dimension of our body, for example, representation of somatotopic, or the visual information in a map, it's not possible that every different type of neuron have a signal, a molecular signal, and the target also. So then this axle will guide to the right place. So Sperry proposed that there must be something much more simple, much more economic, and that's concentration gradient. And he was right. And this is what I would like to illustrate. So look at this. If we see an object, a chicken, on the retina, we have an inverse picture, and then this picture goes to the tectum, the optical tectum, at least in the animal. This is the locus ultra, the colliculus superioris, okay? And the image is again in a normal position, the chicken is there. Now what's very noticed is that here we have the retina, okay? The retina which receives all the images in our eye, okay? We have the nasal part of this retina, and the temporal and nasal. Okay, imagine a circle. Okay, and what's very noticed is that the temporal retinal ganglionic cells always project to the anterior part of the collicle. They never go further, posterior. And the nasal part will always go much further. It has a longer projection and terminate in the posterior part. The Sperry then asked the question, what happens if I cut the retina and turn 180 degrees, okay? So he turned the nasal will be here, the temporal here. What happens with this projection pattern? That is temporal always short, nasal long, okay? Whatever Sperry did, turn 90 degrees, the temporal cells always projected to the anterior part and the nasal projected much longer. So based on this observation, Sperry proposed that there is a chemical affinity. Somehow this says something. And it turned out like this. Okay, look at this. It turned out that tactile neurons, okay, the target, produce the receptor. No, I'm sorry. The tactile neurons produce the ligand, which is called ephrine, in different concentration. And the optic nerve with the growth cone, which arrives into the tectum, will produce the EPH receptor. Okay, remember, ligand and the receptor. When the ligand is a sufficient high concentration, or the receptor is a sufficient high concentration, this will induce through small GTPases a collapse of the growth cone. The growth cone will turn away or stop completely. Look what they figured out. Remember I showed you nasal neurons in the retina, they project always long into the posterior. Temporal always project short. They figured out somehow that in face of this repulsion, ephrine signaling, the temporal neurons has a very high sensitivity, much higher than the nasal. And they figured out that this is because they produce a higher concentration of the receptor. Okay? And when we're getting closer to the temporal part of the retina, the concentration of the receptor will be higher and higher and higher. And finally, in this part, this is the highest concentration of receptor in the membrane of the growth cone. While here, in the nasal part, the receptor concentration is very low. And look at in the tectum. They figured out that the strength of the repulsion is very strong in the, very low in the anterior part. Very low, okay? And it's increasing progressively, step by step. Chaque 100 micrometers is increasing the concentration of the ligand, ephrine, in the membrane of the collicular neuron, okay? And they showed that there is a progressive increase of the ephrine ligand. So here is the explanation and showing that Sperry was right. What happened is that the high concentration of this high concentration of receptor in the temporal neurons in the retina, they are very sensitive and catch even the lowest ligand concentration and they will stop. And just look at this. In fact, this is in the concentration gradient. Neurons here, which has a lower receptor concentration, can a little bit further and so forth. Okay, the nasal will go even further because it has a very low concentration of the receptor. And finally, even nasal axons will be stopped progressively, each axon at a different distance, because of the increase of the ligand concentration. Now you realize something, that one single molecule, a ligand, and one single molecule, a receptor, And change concentration can establish a whole topographical map based on the gradual changes of concentration. So each axon has a sort of target place based on this combination of ligand and the receptor. Meaning that I cannot imagine a more economic system. One single ligand can establish a 2D distribution of axons in the follicle. And the same thing is the cortex. So making topographical maps, organized topographical maps of our body or the outside world, in a way, can be regulated during development by the concentration gradient of guidance factors, receptors and ligand. Okay, this was the story today.